Hello everyone. In this video we are going to continue our discussion about food acquisition in animals. So we are going to focus more specifically on absorption and elimination. So absorption is the uptake of nutrients by body cells, and elimination is the passage of undigested nutrients out of the digestive system. So not all the food materials will be digested. For example, while we can digest different types of sugars and carbohydrates, we cannot digest the fibers. So fibers will be undigested, and for that reason they will be passed out of the digestive system through the anus. We have to keep in mind that the undigested material, they are not universal. So some other animals, they may be able to digest the fibers. So the undigested material is something that may vary from one animal to another. So this figure is a rough diagrammatic figure showing the mammalian digestive system. So as we said, we are focusing mainly here on the mammalian digestive system, and this is a rough figure. It is not an anatomical figure, but to show you roughly how the different parts of the mammalian digestive system connect and work together. So the mammalian digestive system consists of the alimentary canal. So this is the part shown here in white. So this is a canal through which the food will pass through the mouth and will go through all the way through the alimentary canal to be released to be eliminated at the end through the anus. In addition to the alimentary canal, we have accessory glands. So these are the glands shown here in purple. So these glands, they secrete digestive juices through ducts. So we have a series of ducts through which the accessory glands will secrete their juices. These accessory glands include the salivary glands, the gallbladder, the liver, and the pancreas. So they secrete their juices into the alimentary canal through the ducts, and they will help in the digestion of foods. So starting with the oral cavity, pharynx, and esophagus. So the first stage of digestion, the mechanical digestion, will take place in the oral cavity. So by the help of the teeth, we are able to break down the food molecules, the food material, into smaller chunks of food through the mechanical digestion. Then in addition of that, we have enzymes being secreted in the saliva in the oral cavity. One of them is the salivary amylase. So to define this enzyme, this is an enzyme that will break down carbohydrates. So salivary amylase is a specific enzyme acting on the carbohydrate molecules. And as you remember, we said the macromolecules we consume are composed of carbohydrates. We have proteins, we have nucleic acids, and we have fats. So the salivary amylase will specifically work on the carbohydrates, breaking them down into smaller sugar molecules. And in addition of that, we have in the oral cavity, we have the saliva, which help to lubricate the food material. So without the saliva, remember, we are going to pass the food material into the pharynx and esophagus. So if it is not moistened and lubricated with something, it will going to stuck. So in order to pass it through the pharynx and esophagus, one function that is happening in the oral cavity is the presence of saliva, and the lubrication happening there is also an important thing. In addition of that, the saliva contains mucus. So mucus is a viscous mixture of water, salts, cells, and glycoproteins. So again, this will aid in the lubrication process. It will help protect the mouth lining from abrasion, and it has glycoproteins. So many of these glycoproteins are of antimicrobial activities. So all of these are happening in the oral cavity. So to summarize it, number one, the first major thing happening there is the mechanical digestion, breaking down the food by the help of the teeth. And number two, that we started to break down carbohydrates in specific. And this is through the action of salivary amylases. So the food will then be passed through the different parts of the alimentary canal to the stomach. In the stomach, the food will be stored for a little bit of time. Then the stomach will secrete the gastric juice, which will convert it into what we call acid chyme. So the acid chyme is the chyme is the mixture that is made from the food in addition of the acid that is being secreted by the stomach. So this mixture is called the chyme. And if we go through the anatomical features of the stomach, so this is the interior surface of the stomach, what you can see here, and it has these gastric pits. So these are like pores, and what will be secreted through these pores will be coming from the lumen. So we have different cells here, as you can see. These are lining the interior side of the stomach. They will secrete their secretions. It will be released to this lumen, and it will be collected and then released out through the gastric pits. So it will be released out. It will be mixed with the food material, and that's what will compose the chyme. So what are the secretions and what are the cells that do we have lining the stomach? Number one, we have mucus cells. And so these are shown here in red. They secrete the mucus, which we have already mentioned in the previous slides. So the function of the mucus and the composition of the mucus. Number two, the Schiff cells. So here, shown here in pink, and we have the parietal cell, which is shown here in yellow. So the mucus cells, they will protect the stomach lining. The Schiff cell, they will produce the pepsin, so the pepsin enzyme. And number three, the parietal cells, they are going to secrete the hydrochloric acid, HCl. So parietal cells, they produce the HCl, the hydrochloric acid, which is very acidic. So we have to protect the lining of the stomach by mucus. So simply, this is the mechanism through which the stomach does not, or the acids of the stomach does not digest the cells of the stomach themselves. Then we have Schiff cells, which produce an enzyme called pepsin. So as you can see here, the parietal cells will produce the HCl, the Schiff cells will produce the pepsin. Actually, pepsin is secreted in inactive form called pepsinogen. This pepsinogen will be converted into the active form of the enzyme, which is called pepsin, through the action of HCl. So HCl secreted in the parietal cell will help the conversion of pepsinogen, which is inactive, into pepsin, which is active. Then more details about the secretions of the stomach. So the acidity is very low. So we have a pH reaching about 2, which kills the bacteria and denatures protein. So 2 functions, or even like 3 functions now. So we said that number one, the HCl helps converting the pepsinogen into pepsin. Number two, the very low pH of the HCl makes it antimicrobial, so it kills most bacteria. And number three, it helps in denaturing proteins. So proteins are composed of 3-dimensional shapes of the amino acids. So amino acids are arranged in 3-dimensional structures, and by the release of the acid, this will cause this structure to be lost. This will cause this folding to be unfolded. So now we started to act on proteins in the stomach. So the gastric juice is composed of 2 things. The gastric juice is composed of the HCl, the hydrochloric acid, in addition to the enzyme, which is pepsin. And both of them, they work together to cleave protein. So cleavage of proteins will happen in the stomach by the aid of two things, the HCl and the pepsin. So as you can see, in the stomach we are mainly talking about digestion of proteins, while in the oral cavity we mainly focus on the digestion of carbohydrates by the action of the salivary amylase.